All right, so we're just going to start with osmosis, kind of where we left off yesterday. Osmosis is a kind of diffusion. It's a diffusion of water. And since it's a kind of diffusion, water always moves from what to what? Remember, high to low is not good enough. You got to say high concentration of water to low concentration of water, which means it's passive or active. Passive. Then it's powered by heat in the environment, thermal energy in the environment, just regular old movement. But a lot of times we don't talk about osmosis in terms of the water. We talk about it in terms of what's dissolved in the water, and those are solids. And where does water want to be in terms of solids? Where there's more solids. So water moves from where there's low solute concentration to where there's high solute concentration. That's always the case. Another way of saying that is saying it moves from hypotonic, which means less solutes, to hypertonic, which means more solutes. All right, the other thing that I want to focus on today, though, is this water potential bit. So let's talk a little bit about water potential. We'll put some equations up here first of all. What's the equation for what's the symbol for water potential? Okay, it looks like a trident. It's actually called what? What's that little symbol called? Psi, PSI. And what's the little t there mean? Total. Remember, water potential takes into account two things. What are they? Solutes and pressure. So we call the solute potential plus the pressure. Remember, we also had another equation. Anybody remember what that equation was to calculate the solute potential? Negative I CRT. Where I was what? Ionization constant. C was concentration of solutes. R is the gas constant. And big T has to be what? Kelvin temperature. Before we get into a problem, water always moves which way in terms of water potential? Higher to lower. So let's first of all say we had a beaker. And let's say in that beaker we had 0.6 molar. And let's say it's calcium fluoride. Now let's say this beaker's open. This is not a top. It's just a rim of the container. It's open. And let's say the temp inside there is maybe 20 degrees C. And then we're going to take a cell and put it in there. And let's say I told you that the water potential inside the cell was, let's say, negative 2.5 bars. That's inside the cell. And I want to know, first of all, what's the water potential in the beaker? And then I want to know which way is the water going to move. Is it going to move into or out of the cell? So first of all, that beaker's open. So what's the pressure like in the beaker? It's just atmospheric pressure, which is the same as what's acting on the cell. So we can just say the pressure is 0 when it's open like that. So this part we're going to say is 0 to begin with. Which means we need to calculate the solute potential. What kind of chemical is calcium chloride? It's got a metal in it, so it is in... What kinds of chemicals have metals in them? Salts, which are held together by what kind of bonds? Ionic. So the first thing we've got to do is figure out this ionization constant. And we just take our chemical and we disassociate it. We break it into its ions. What's that going to break into? Ca2 plus and 2Cl minus 1, right? What's the ionization constant of that? 4. you count up the total number of ions given off from one unit of this. So 2 positives, 2 negatives, 4 total charges. It's an ionization constant of 4. So far we're at -4. The C is the concentration, so that's the 0.6. And remember that big M, molarity, actually stands for what? Moles per liter. Moles of solute per liter of solution. All right, the constant. Anybody remember what the constant is? Yep, 0.0831. And the units are a little weird, but it's bars. And this, by the way, this constant is given to you on your formula sheet. Bars times liters over moles times Kelvin. And then the only thing we've got left is that the Kelvin temperature, which would be what? 293. Celsius plus 273 to get Kelvin. So first of all, we check all our units, and moles will cancel out right here. Liters will cancel out right here. And kelvins cancel out right here. So we're left with bars. Now we need to calculate it out. So 4 times 0.6 times 0.0831 times 293. All right, and we end up with a solute potential of -58.4 bars. And we're trying to find the total water potential inside the beaker. The pressure is 0. The solute potential is -58.4, which means the total water potential in here is -58.4. So which way does the water go? Into or out? Out. -2.5 is bigger than -58.4, right? It's a bigger number. So the water moves out. And if that's a cell with a wall, what do we call what's going to happen to that cell? Plasmolysis. Shrivels, right? If it doesn't have a wall, if it's like an animal cell, what do we call when it shrivels up? Crenation. Now, let's say the water was going the other way, though. Let's say that the -2.5 was out here, and the -58.4 was in there. What would we call what happens to the cell if it's a cell with a wall? It'd build up turgor pressure. It'd get stiff, turgid. And on the other hand, if it was a cell without a wall, what would happen? It would burst, it would lyse. Everybody okay with this water potential thing? Water moves from high water potential to low water potential. Be able to use those two formulas. One thing to think about here with this I. If the compound is not ionic, if it's something that's just made out of non-metals, like a sugar, what are you always going to plug in for I? 1. Don't forget that negative. That negative is important. But if it's not an ionic compound, if it's not a salt, plug in 1. If it is a salt, it's got a metal in it, you got to do this. Count up the total number of charges, and that gives you your ionization constant. Everybody good with that? A couple of things to think about here. You put salt on a slug, what happens? He shrivels up because his membrane is what? Permeable to water, but not permeable to the solute. So the water moves out from higher concentration of water to lower concentration of water. Remember, when you're talking about osmosis, water tends to move, but what can't? But the solute can't. That's an important thing to think about. What was a contractile vacuole? Yeah, it's a thing that it's usually protists is usually what we talk about. I mean, protists, most of them are living where? What kind of water? Fresh. And inside that produce body are salts and sugars and things, right? So what's their issue with water? It keeps going in. And if they didn't have a way to deal with it, what would happen real fast? It would burst, it would explode. So these little contractile vacuoles are sort of like pumps. They collect the water, pump it out. Collect the water, pump it out. And they're constantly doing that to keep that cell from swelling up and popping. Morning. There you go. I'll just leave it pop open. All right, guys, let's go a little further. Here's a picture, and this doesn't show up very well in this projector, but this is what a normal plant cell ought to look like. Because most of the time, a plant is soaking in what kind of solution? Compared to its insides. Mostly hot photonic, right? It's soaking in fresh water. And when we say fresh water, the water out of the ground, is it completely absent of solutes? It's got all kinds of stuff in it. But still, the plant has more inside it than the groundwater does. So the water is always going to move which way? Thank you. If there's more solutes in the plant cells and less in the groundwater, water moves which way? Into. And that's a good thing for the plant, right? That's one of the reasons it gets water in the first place. So the plant cells normally all spread out and it fills up the cell wall and there's a lot of turgor pressure in there. It's turgid. But if all of a sudden you start soaking it in salt water, which we did, the water comes out from hypotonic to hypertonic and it shrivels, and we call that plasmolysis. There's some more plasmolyzed cells. And there's what it looks like in an animal cell. That's a red blood cell. Here it's in an isotonic solution, which means the concentrations inside and outside are the same. That's what you want. When things are in isotonic solutions, is diffusion still happening? Yeah. Just know what is happening. No net diffusion. Here the cell is in a hypertonic solution. So the solutes can't move because they're ions. So the water's moving, and the water's moving out, mostly, from areas of less solute to areas of more solute. And notice there's some arrows showing water going in. Will some water go back in? Yeah. But the net movement is always which way? Out. Less solute to more solute. And here we've got one that's going to swell up and burst because there's more solutes on the inside than there are on the outside, and water's going toward those solutes. Now, if those solutes could move, what would happen? They could move. If the solutes could get out, what would happen? They would, and then the cell would be okay, right? But in this case, it can't because they're probably what kind of solutes? They're probably either big or charged, and those have a hard time getting out, right? They're stuck. Let's see, we got our water potential stuff. So just some reminders about the other kinds of transport. Facilitated diffusion means diffusion with a little bit of what? Help. And what provides the help? Yep. What's the name for the proteins? We might call them transport proteins. I usually call them channel proteins. And these things help to move big or charged things, bigger ionic compounds. But they're always moving in which way? High to low, with the gradient. So this is a passive process. It doesn't require any energy used by the cell. It's powered by heat in the environment. This is an important way for cells to move things like sugars and amino acids and stuff like that. Another kind of moving that we've talked about is active transport. And we know that active transport always moves things against the gradient, which means it's moving from low to high concentration. Active, if you ever see that word in biology, it always means what? Requires energy, and let's be more specific, it requires the cell to use what? ATP, its own energy source. ATP has to be hydrolyzed. Hydrolyzed means what? broken down to give off that energy. And by far the most important example of active transport in your body is the sodium potassium pump that we've mentioned several times in here. Mostly it's found where? In nerves. And the job of that sodium potassium pump is to basically build up a gradient, sort of like what we've been talking about in cell respiration and photosynthesis. It's constantly pumping potassium ions into the neuron. So a big gradient of potassiums is being built up inside here. And it's pumping sodiums out of the neuron. What do those sodiums want to do? They want to go in, right? What do the potassiums want to do? They want to come out. But they can't because they're both what? Ions. So this is a way to store energy, to store potential energy. And there are little gates on here, little channels that will open up. And when they open, what happens? Those things move. And since they're ions and they're moving, what just got generated? Electricity. And that's the way your nerves make electricity. The process of pumping sodiums and potassiums uses more ATP than anything else that your body does. Seems kind of minor, but without that, your brain wouldn't work, your heart wouldn't work, nothing in your body would work. So it's pretty important. All right, a couple other reminders about cell moving kinds of things. Endocytosis. Literally means what? You break it into pieces. What does it mean? Yeah, sort of. Cyto means cell, and endo means into. So this is taking things into the cell. But when we're talking about endocytosis, we're talking about taking in big things, big amounts of things always. There's two kinds of endocytosis, phagocytosis and pinocytosis. We talked about both of them several times. Well, let's go to a picture here. Here's phagocytosis. Notice there's a typo up there. It's not phagocytosis. You could get phagocytosis if you did a lot of phagocytosis, I guess. Right? Right. Anyway. I mean, the cell is surrounding the object. It's pinching in around it with its cell membrane. And eventually this side of the membrane is going to touch that side. What happens when phospholipid and phospholipid touch each other? They fuse. They make a sac, a vacuole. And that thing's now enclosed in like a little baggie. Goes into the cell. And what might that be that I'm drawing that moves up there beside it? The lysosome. And my arrows here would represent what? It's pumping enzymes into that little vacuole, and he's going to break it down right there in the cell. And what do we call that process? Intracellular digestion. Breaking it down. And that's phagocytosis. Pinocytosis works essentially the same way. But instead of taking in solids, it's taking in dissolved things. Solids mostly. Here, you can see it's surrounding the solutes. These are little dissolved particles. Put them in a vacuole, and then it will break them down from there. Occasionally, some types of pinocytosis require receptors to trigger them to happen. So you might see pinocytosis called that, receptor-mediated endocytosis. And notice what's happening there. The solutes are binding to these little receptors. And when enough of them bind, that tells the cell it's time to pinch in. It's time to imaginate, pull this stuff in. But those two things, phagocytosis and pinocytosis, are ways to take in big amounts of stuff. Are they active or passive? That takes energy, right? To do all that pinching and moving by the cell. Those are active processes. That requires ATP to be used. All right. Any questions on the cell transport business there? We're going to talk a little bit about cell signaling. All right, if you remember when we first started talking about cell signaling, we watched a Mr. Anderson video, and he made all these analogies about different ways people communicate. Anybody remember what his analogies were? Sticky note, Facebook, and yeah, email. And he compared those to different ways cells communicate. One of those ways that cells communicate are cells that are side by side, what we're going to call local signaling. And remember that there's a lot of different ways cells can do this. But one of the ways is just by the connections between the cells. Let's say we had this is a plant cell, and here's another plant cell right beside it. They're adjacent cells. What could get in the way of them communicating with each other? A big old wall. But what actually exists are like little tunnels between the walls. What is that kind of connection between plant cells called? Look up here on the board if you don't remember. Plasmodesmata. And those little tunnels between the cells let them transfer information and actual materials. It's a type of communicating junction between cells. What's the prefix for between? Inter. So we might call that an intercellular junction. The junction between the cells. Animals have a very similar kind of connection between their cells. We kept talking about the connection between like heart muscle cells, cardiac muscle cells. What do we call that kind of junction that lets animal cells communicate with each other? Yeah, junctions. Should know those terms. We also spent some time talking about the immune system and how it communicates. And what we're looking at here, let me get my bearings here on what's going on here. All right, this would be like a white blood cell that eats stuff, phagocytizes stuff, and you see it happening here. Anybody remember what we call the main kinds of phagocytic cells in your immune system? Macrophages. So this would probably be like a macrophage. And it's eating some kind of germ right here. Let's just say that's a little virus. So it phagocytizes it, puts it in a vesicle, and then does what to it? What's the word though? When it breaks it down inside itself. Intracellular digestion. Breaks it into pieces. And most of those pieces, the cell just reuses the amino acids and the nucleic acids and stuff like that. But a few of the pieces get put where? Yeah, out here. This little triangle right here is a piece of that virus. What do we call proteins from something that's not supposed to be in you? Antigens, right? So that's a foreign antigen, a piece of that virus. And what's the little green thing called? Yep, it's called an MHC class 2 protein. And I told you to compare that to like a trophy hand. That cell is saying, hey, this thing was in our body. I killed it. I kicked its butt. Here's a piece of it. But there's more of them. You need to go do what? Hunt them down and kill them. And who's it telling that to? Who's it trying to communicate? This cell that's connecting up to that, and they have to bind. What do we call that cell that binds to this macrophage in its class 2 MHC? Back up a step. They're called helper T cells. And basically, this macrophage is showing that helper T cell what this germ looks like. And then what will the helper T cell do once it gets activated and becomes aware of the germ? It sends out all kinds of signals to all parts of your immune system, and it activates them. So the cells that have a class 2 MHC are only the immune system cells that are like phagocytes that eat things. Macrophages, there's some other kinds, but the phagocytic cells have those. The other parts of your immune system, or actually all of the cells in your body, have something similar though. What do we call that one that every cell in your body has? It's called the class 1 MHC. And essentially, your cells are taking what from inside and putting out there? Yeah, little proteins from inside the cell. Most of the time, what's going to end up out there on that class 1 MHC? It's the normal protein from inside the cell. Who's coming around checking those out? Different kinds of T cells, helper T cells, killer T cells are checking them out. And if it's a normal protein, life is good. They leave that little cell alone. Everything's the way it ought to be. But let's say that cell, this particular cell, has a virus infection. Where do virus infections do their damage? Inside a cell, right? They're intracellular parasites. They got to be inside. So when the cell is sticking up proteins, what's probably going to end up on some of those MHCs? The virus proteins, antigens. And if this is a helper T cell here, and it binds to that virus protein, it realizes it's a virus protein. So number one, it's going to send out signals to other immune system cells saying, hey, this thing is in us, we need to kill it. But it's also going to tell what cell to come visit over here. Yeah, killer T cell or cytotoxic T. And essentially, that cell's going to go Wolverine on this cell, punch holes in it, and all its guts are going to leak out and it's going to die. With the hope that it kills it before what finishes inside that cell? The lytic cycle, right? Which is the virus reproduction. Because if it doesn't kill it before that happens, it's a little too late, right? Because the viruses are going to come out too. Does that make sense? Everybody okay with that? So that's local signaling, when cells actually have to be side by side and connect to each other to make it work. And another thing about your immune system, remember there's two main parts to what we call your specific immune system. There's a whole different part of your immune system called the non-specific part. What would be the most important part of your non-specific immune system? This is the most important part of your immune system in general. Your skin. That's pretty important, right? Keep stuff out. Your mucous membranes, like in your nose and your mouth and all the openings in your body, those are pretty important. They keep things out. But they're not specific to a particular germ. These things are specific. Now, we just talked about both of these a little bit. You got an antigen. It gets eaten by a macrophage. What do we call it when that macrophage takes those antigens and sticks them out there and starts showing them to other cells? Antigen presentation. So that's what's going on here. Antigen presentation. And that's telling that helper T cell, hey, this is in, let's go find it, go kill it. That helper T cell stimulates two important immune responses. One of them is it tells these cells called B cells what that germ looks like. And the B cells eventually turn into what are called plasma cells. And they start chugging out antibodies. What's an antibody? What do antibodies help you do? Kill germs, okay? Whenever you hear the word antigen, I want you to think the word antibody. Because antibodies are made against what? Specific antigens. And they either help you to kill it or help you to find a way to get rid of it. One of the two. But they're specific to that germ. Notice that most of the B cells turn into plasma cells and start making these antibodies. Millions of them, like per second. But a few of the B cells hang around as what are called memory cells. That's important. Because if you get that same infection in you years later, what do these do? What do the memory cells do? This process that we're looking at up here on the board, these first few steps take a long time, 5 or 6 days to happen. But if you already have memory cells, how long does it take your body to respond? It's like instantly. So fast that what doesn't happen? You don't even get any symptoms. Notice there's also memory helper T cells that get formed. And then this response over here is the one we were talking about a second ago. This is called cell-mediated immunity. This helps you deal with your own cells that are what? That are sick. Those antibodies can only get to germs that are where? Outside the cell, because the antibodies are proteins, right? They're big. They can't get into a cell. But they can help you kill germs that are in the blood or in the body fluids. But they can't help you when the germ's actually already inside the cell. That's what this does. This deals with germs that are already inside your cells. And this is where the cytotoxic T cells recognize that your own cell's sick, and then they kill it. Notice there's also memory cells that form there. So these memory cells are just what they say they are. You've had a germ before, you fought it off, you remember what it looks like, and if it gets in you again, you get it fast so that you don't get sick. That's a good example of local signaling. Another good example of local signaling is the way nerves work. Let's talk about these nerve parts. These pieces called dendrites up here. Dendra means like a what? Like a tree. So it looks sort of like tree branches. What do dendrites do? They receive incoming information. Maybe from another nerve or for some kind of receptor. Incoming stuff though. That information then goes into the main part of the nerve here, what's called the cell body, or sometimes it's called the soma. And that's where the nucleus is at, of the cell. And then most neurons have one long extension coming out right here, and that's called the axon. And that sends information out. It usually has this fatty coat around it called what? What's this white stuff in the diagram represent? It's like insulation. It's called myelin. I mean, essentially that axon is like what human machine? Yeah, it's like a piece of wire. Do you want a naked copper wire laying around if there's power running to it? Not a good idea, right? And so you don't want your nerves to not have insulation either. And that's pretty much what the myelin is. It's like insulation around those nerves. Eventually, though, this nerve, the one on the left, is going to connect to another nerve or some kind of muscle or something. And what's that connection called? It's called synapse. Do they actually touch? They don't. There's a little gap, and that little gap is actually what's called the synapse. Let's look at it in a little bit more detail here at the synapse. So here we're looking at the end of an axon, and then this would be the beginning of the next neuron, the dendrite on the next one. So we might call this nerve up here the presynaptic neuron, and this one down here is postsynaptic, after the synapse. The way they communicate is that you've got electricity running through this nerve up here, the impulse. And when it gets near the end, it causes this presynaptic neuron to release what? What do these little red dots represent? Neurotransmitters, little signals. What word can we use for any kind of cell signal that can't go into a cell? A ligand. So they're examples of ligands. Signals. And typically for a ligand to work, it's got to have what to go with it? A receptor. So on the postsynaptic neuron, you see these little protein receptors. And those are a specific kind of protein receptor. They're called ligand-gated what? Ion channels. And when enough neurotransmitters bind to these little receptors, what happens to those ion channels? They open. And when they open, what rushes into this postsynaptic neuron? Sodium. And that's moving ions, which is a stream of what, essentially? Electricity. So the impulse continues. But that's another example of local signaling. Cells that are side by side communicating via some kind of ligand. All right, let's see. Last thing that we'll look at. This was a question on your test, which you guys in B class will get back today. These 3 stages of cell signaling. It will show up on the test, the midterm, I promise. When cells communicate, step 1 is reception. And that's essentially that you have a ligand and a receptor bonding together. They have to fit sort of like what? A lock and a key. And when they fit together, what physically happens to that receptor? It changes shape, conformational change. And that typically, if we're talking about an extracellular receptor, most of there's part of the receptor on the outside, but actually it extends all the way to the inside, right? And when that conformational change happens, usually what part of that receptor is actually changing? The inside part. And that somehow triggers step 2, which is transduction. Transduction is the complicated part of this process. But I told you to think of 2 words when you thought of transduction. What were they? Amplification and translation of the message. So, first of all, usually the signal, that original signal, the first message, triggers the creation of what? Think about the whole epinephrine thing. What was the first thing that really got made inside the cell? Yes, some kind of second messenger, CAMP. And a lot of that got made. And the job of that second messenger is to spread that message throughout that whole cell, right? We're not talking about from cell to cell, we're talking about inside that particular cell. It spreads it. And the CAMPs help to activate certain kind of protein kinases. And those turn on other proteins, and those turn on other proteins, and those turn on other proteins. We call that what kind of process? Yeah, phosphorylation cascade. Until eventually, the protein that you're trying to turn on gets turned on. In the case of the liver cell, what were we trying to turn on? The liver cell responded to adrenaline. What were we trying to turn on? We were trying to turn on enzymes that break down glycogen and therefore release glucose. And that's the response. So the last step in the cell signaling process is the response. And usually it's either when a reaction gets turned on or off in a cell, or maybe it's where a gene gets turned on or off. Make sure you know those 3 stages. Make sure you can explain sort of what's going on in each one. Well, the midterm is going to be all multiple choice. So just understand what they are, and you can really answer the question. All right, I will stop this recording, and I will post both videos of yesterday's and today's. We covered different things in the 2. Let's